All right. So animals. There are some overall general features of animals that we'll discuss. Yeah, I know. The first is heterotrophy. As we all know, heterotrophy is breaking down of organic molecules to get our energy, which is in contrast by everything else we've seen thus far in the past couple of weeks, where we were talking about plants, where they in fact get their energy by photosynthesis, where again, our friends here get them by heterotrophy. This is where we fall into the category. Now, we are all multicellular, so all animals, as we'll discuss, have multicellular aspects to them. So protists clearly are not animals because they're multicellular, but plants are not animals because they're clearly not heterotrophic. There's no cell walls, we have active movements, and there's a wide diversity of form, whereas most plants, as we discussed, fall into basically like 3 categories. Right? We have the bryophytes, we have the vascular plants, and the seeded plants. There's those standard forms. There's a much wider variety of forms here. Now, there's also a wide diversity of habitat. There's about 35 to 40 phyla that we'll discuss. Sexual reproduction is essentially a must here. There's very few animals that don't reproduce sexually. There's also tissues. You know, we talked about plants, they have different types of tissues, like ground tissues and merostems and all that fun stuff. But our tissues are organized into what we'll turn to call as organs, as we kind of all know. And then also there's embryonic development. So we have development of actual embryos, not like the plant embryos, but actual embryos. And we have the diversity of life, ranging from the beautiful to probably the things you don't find so attractive, but I really like, because who would not like a blue centipede? Looks pretty cool. And then stuff that you, again, developing as embryos. So lots of diversity to cover. And again, I think you guys will like this much better than plants, as most people tend to do. Now, there are 5 key innovations that we're going to walk through: symmetry, tissues, body cavity, patterns of development, as well as segmentation, all giving us aspects of animal biology. So symmetry is a really interesting concept. Now, if you think about what symmetry is, if you cut yourself in half vertically, you're symmetrical from side to side. If you cut yourself horizontally at the waist, you're not symmetrical from top to bottom. That's what we're talking about here. Is there a mirror image of you? Now, in the case of sponge, we'll talk about sponges first, but we're not quite there yet. They lack a definite symmetry, but basically all other animals have a symmetry we define as being radial or bilateral symmetry. Now, radial is like, think of like a pie, right? A pie is radial, that symmetry all the way around, provided it's not made by someone who doesn't know how to make a pie. Like if I make a pie, my pie is not radially symmetry because I can't make a pie. My wife can, but I can. Then we have bilateral symmetry. This is what we're divided into. So the things that we commonly think, like birds and snakes and grasshoppers and fish, all have bilateral symmetry. But things like sponges, sea cucumbers, pies clearly not being animals, of course, but they have that level of radial symmetry, that 360-degree symmetry around a central point. And again, we can see what this looks like. Radial symmetry, this is a hydra. We can see here on the top, we have radial symmetry. It's the same all the way around. Whereas we look at our friend here, the sea turtle, it has an anterior, the front, and a posterior, the back. And you have a line down the middle, which we call the sagittal plate. And this sagittal plate gives the turtle bilateral symmetry on its left and right side, however you want to call it. Now, what are some advantages of bilateral symmetry? Why do we have it? Why is it a really successful type of symmetry? Most of the animals we're going to look at have this type of symmetry. So why is it so successful? Well, a couple advantages here. We have what's called cephalization, which is the evolution of a definitive brain area. Obviously, the brain is awesome. This is how you can sit here and listen to me talk, how you can write, and do all the things that a brain allows you to do. But if you do not have bilateral symmetry, brains are not exactly the most common thing, as we'll see. You can also have directional movement. If you are symmetrical fully round, you really don't have a way to propel yourself forward in a meaningful fashion. You don't really have the physics to sort of make it through to point A to B. But with bilateral symmetry, I have that capacity to propel myself forward. A grasshopper has the capacity to power itself forward. Now, tissues, as we mentioned, are another big aspect. So we start off, as we did with plants, we start with zygotes, which are just fertilized eggs, which then give rise to other cell types. So we actually call these totipotent, which I think is a fun word to say. And basically, as the embryo develops, as the zygote continues to develop, and you guys probably have a general idea about this, but as you develop from an egg, you specialize into different tissues. Now, this process of specialization is irreversible. So once something becomes brain tissue, it is brain tissue forever. Now, there are special types of tissues, as you guys probably know. They're called stem cells. We talked about plant stem cells quite a bit when we talked about apical meristems, right? Those are just the capacity of plant cells to differentiate into any type of plant tissue. But as you guys know, stem cells, from an animal perspective, give rise to different tissue types. So a stem cell for us can differentiate itself into liver, kidney, brain, and so on and so forth, which is why they show a lot of medical promise, right? If I lost an arm, well, if I have a tissue that can differentiate into a brand new arm, kind of a big deal, right? So that's why stem cells are so awesome. As I mentioned, this process is irreversible, except for our lovely sponge friends. We'll learn lots about sponges, weird group of organisms. They're also pretty unremarkable. But sponges, which are defined as actually the simplest animals, they don't have distinctive tissues, nor do they have distinctive organs, unlike the rest of the things that we'll look at today. But what they do have is the capacity to disaggregate and then reaggregate their cells together. Now, what does that mean? Think about if you were blown up. I know that sounds awful, but picture you were blown up. If you were a sponge, your process to reaggregate your cells means all your cells could come right back together and form you again. Which is wild, right? I always find that really cool. So that's our sponges, but all other animals have distinct tissues, irreversible cell type differentiation. Now, the body cavity is another important aspect of our biology here. Most animals, they have embryos. Excuse me. Sorry about this. I don't know why. They produce 3 distinct layers, which makes them what we call triplobasic. So the first layer is the outer ectoderm, which is the body covering, as well as the nervous system. We have the middle mesoderm, which will be our skeleton and muscles. And the third is the inner endoderm, which is our digestive organs and intestines. So these are our layers that we have. And all animals that have these distinct layers have bilateral symmetry. Now, our Nadarians, which we'll look at later on, so this is sponges and corals, they are actually called diploblastic, which they actually only have an ectoderm and an endoderm. They don't have a mesoderm. And that will become clear when we take a peek at Nadarians. Kind of a weird group of organisms, as a note. And then the other thing is sponge lack all these things. So sponges will always be kind of our contrast for what we're looking at, because again, they're the most simple of all organisms. Now we have the body cavity, which is simply the space surrounding that mesoderm tissue that's formed during development. And there are 3 basic body plans: the acoelomates, which have no body cavity, the pseudo-elomates, which have a body cavity between the mesoderm and the endoderm, which is what we call the pseudocollicule. I'm going to try. My pronunciation is already off today. That's not good. And then we have the chelomates, which is the body cavities entirely within the mesoderm. That's what we have. These 3 distinct body planes. And we can see what these look like. So we have a planaria, or a flatworm, here at the top. We can see its ectoderm. We can see its endoderm. We can see the digestive cavity here. And then we have this mesoderm-derived tissue. Then we can look at a roundworm here as an example of our second one, where again, we have the ectoderm tissue. We have the endoderm tissue. We have the digestive cavity. And we have that sort of inner space as well. And then we can look at an annelid as an example of our final group. Again, we have that ectoderm, we have that endoderm, we have the digestive cavity, and then we have that, again, that inner space. So overall, 3 distinct body plans that we do see. Now, circulatory systems, as you guys know, kind of these needs, right? Circulatory system meaning how you move liquids, how you move items around your cavities. Plants, that was xylem and phloem, right? In veins, within leaves, right? In us, in other things, there are 2 distinct types of circulatory system: open or closed circulatory system. Open is when the blood passes from vessels into the sinuses, then mixing with body fluids and re-entering the blood vessels. That's an open circulatory system. Now, when we have the closed circulatory system, blood moves continuously through the vessels that are separated from the bodily fluids. So again, 2 distinct types of circulatory systems we'll see across different organisms today. And we also have different patterns of development, and evolution has taken a wild and wacky turn here there's way cool ways things develop. It's really cool. Developmental biology is a really fascinating field. But all development is derived from mitosis, right? So there's the division of cells, as we all know. But basically, there's basically two ways we have to develop what we call a bascula. It's basically just like a hollow ball of cells. Now, there's two ways that this develops into a blastophore, which opens to the outside, or archaeopteron, which is a primitive body cavity, again, more so what we have. So again, two distinct ways for things to develop. So this leads us to two groups, large groups, like really large groups. This is like one of the highest levels of grouping for animals that we will see. We have the protozoans, which develop first from the I always forget this one. So from the near to the blastophore, so the anus, if it develops, either forms a blastophore or another region of the embryo. And then we have the deuterosomes, which develop the anus first. I know it's kind of a weird way to think about it, but develops the anus first, and the mouth develops later. So we either develop as a protozome, mouth to anus, or anus to mouth. That's the sort of defining feature we have here. I know not the most exciting of features to differentiate things, but again, two distinct forms of development: protozomes and deuterosomes. Now, there are also other things that difference between our protozomes and deuterosomes. First is the cleavage pattern of embryonic cells. For protozomes, they have a spiral cleavage, spiral, like corkscrew shaped. Then we have deuterosomes have radial cleavage, come on top of other previously, I think like the rings of a tree, right? That level of development. The developmental fate of the cell. So the protozomes have determinate development, so the cell fate is determined very, very early. Whereas the deuterosomes, they have indeterminate development, where the cell fate is not determined until after several mitotic divisions. We fall, again, as deuterosomes. And then the final aspect, which I'm just making mention of, I won't really dive into it, but it's the formation of the coelom. Not too important, but again, a distinguishing factor. So this is just a handy-dandy summary table. Just as a note, we have our protozomes and our deuterosomes, how they cleave their cells, what the fate of their embryonic cells is, the fate of the blastophore, and the formation of the coelom. Again, what do you develop first? Your butt or your mouth? How does this opening develop? Does it become a mouth first? Does it become a butt first? I don't know, kind of a weird way to define an organism, but again, two distinct groups. And I told you these are one of the largest divisions on the animal tree of life. And there's a reason for that, because this idea of development is actually relatively important in terms of how you set up a body plan, as we'll see. Now, one of the final aspects we're going to discuss is segmentation. What does segmentation mean? Just having different segments of your body. Right? So I think the easiest way to understand segmentation is we think back to our picture of our centipede from the first slide we looked at. A centipede has multiple body segments, a bunch of different things attached to one another. But segmentation as an evolutionary process does give some interesting aspects. The first is being organ systems in adults, such as we see, lung systems, or copying circulatory systems in annelids, but also allows for the more efficient and flexible movement, because each segment can move independently. Now, I gave you the example of the centipede, but think about your own body. It's segmented. I got arms, I got legs, right? Allows repeating structures to occur, allows autonomy, greater range of movement. And as we'll see, segmentation has appeared several times in evolutionary tracks, but some of our most basic animals that we'll discuss today lack segmentation as an idea. So you'll see when we talk about sponges and a few other organisms, segmentation is not just it. So let's dive into animal phylogeny. Now, historically speaking, we've talked about how phylogenies are made with traits. Like I measure, does it have this trait? Does it have that trait? But we know DNA is kind of the bee's knees for developing phylogenies right now. And so, as I mentioned, we have between 30 and 45 distinct phyla of animals, which is a lot. Now, does that make animals the most diverse thing on the planet? No, it doesn't, right? We talked about our protists, we talked about our bacteria, we talked about our fungi. those are by far the most diverse things on the planet, but animals have been studied pretty dang heavily, right? So just as you know. So let's dive into how we classify animals. Now, animals are divided into distinct phyla, and they're grouped into two larger branches. The first is the sponges, which is a monophyletic branch. They are arguably the first group of animals to pop up on the planet. And as I keep mentioning about them, they're a great outlier because they are very ancient, very, very simple. So our sponges, they're monophyletic, meaning there's only one branch of them coming off, very, very old. Now this leads us into Humatozoa, which is animals other than sponges. So you guys should hear some echoes of protists there, right? Protists are everything but X, right? That's the same thing for Humatozoa, right? Anything but a sponge, which I always find funny. Now we have Nadaria, which actually are really early branching. So this is our sponges and corals, really early branching of our friend Humatozoa. And then we have the Bilateria, which is basically everything else. So this is what we have. So let's look at some of the most abundant phyla that we have out there. So we're just going to give a quick overview, and then we're going to dive into most of these. We have Arthropoda, which is arguably the most diverse of all the animals. Now, depending on who you ask, I know it's kind of hard to see this on the right hand, but it depends on who you ask. This group of organisms has either 600,000 species or like 5 million species. It depends on who you ask. And I say that because there's about 600 to 700,000 described species. People estimate there's, again, well over 2 million species. So it just depends on who you ask. And if you ask me, I'm going to tell you there's over 5 million because, again, super diverse group of organisms. But arthropods are like things like beetles, crabs, lobsters, butterflies, fruit flies, right? All the things that we'll discuss today that have a sweet exoskeleton. Then we can look at mollusca, so mollusks, oysters, clams, gastropods, slugs, octopi, things like that. Chordata, which is where we fall in, mammals, fish, reptiles, birds, amphibians, all those fun things. And then we can look at what we call flatworms with the platyhelminthes. Hard word to say. But there's lots of these as well, really tiny worms. One of the cooler ones is actually planaria. Really cool worm. You can cut it in half and it can regenerate from its halves. It's actually kind of a cool story. So. And then we have some more. Again, take a peek at these when you want to. But again, we're going to dive into most of these today. And again, even more, right? Lots and lots of diversity here. Even more. Ctenophores and so on and so forth. So lots of animals that we're going to discuss, lots of diversity, lots of varying numbers of species. We see some pretty high species numbers, right? A couple millions high. But for instance, for this phyla down here, there's only one species. No. Does that mean there's only one species? No. But right now, there's only one species. So that's our phylogeny. Again, we're going to go into each one of these groups, talk about some of the major evolutionary adaptations here, what makes them different, what makes them unique, and how do we go. Now, sort of some of the major developments in animal phylogeny was actually when the annelids, which are just worms, and arthropods were separated into distinct clades, and then arthropods were subsequently grouped with what we call the exozoans, which basically those animals that molt their cuticles. Now, there are some issues with this. As with every phylogeny, it's drawn based upon the data you have, and our data is always inherently incomplete, right? Always incomplete. Because, how can we possibly know all the organisms? But there's lots of issues, so including Netaphora, so our comb jellies. We don't quite know where they fit. They're jellies, but they don't quite seem to fit where they do. And then we have the Ketognathia, which are arrowworms. Again, not quite sure where they go. There are many, many other issues, but these are actually two of the biggest issues. I know you guys probably don't think about, oh, there's issues with animal phylogenies and debates within animal phylogenies, but these are the two big ones, just as a note. So now we have sort of what we call the telomere. So we sort of discussed this topic just briefly, right? This developmental structure that forms in our groups of organisms. So this historically is actually appears to be a really important aspect of animal biology because it seems to only have evolved once, and all deuterosomes have these, but it's actually variable in our protozoans. So, so it's actually a pretty handy thing to do. But We can further divide our protozoans into the lophotrichozoans, which are grown by gradual addition to body mass and have a spiral cleavage. You can imagine that's where we fall into, right? We just start with a ball of cells and add more cells to it to become human. And then we have exozoans, which are animals that molt, including many arthropods and many other phyla. Now, within our deuterosomes, there's chordates and ectoderma, which are very lacking in species. Not lacking, that's the wrong word. They have less species than their protozoan counterparts, and they're actually more uniform in their ways, despite great other differences in their structures. But this is our modern phylogeny as we know it. So we start off here with Porphyria, which is going to be the most basal of all the lineages. So we start off here with Metazoans. Metazoans being a broad term for any animal. Any animal is defined as metazoan. Porphyria, our sponges, are by far the most basic of, or basal, I should say, of all our animals out there. Now you'll notice that chanoflagellates, our protista that we talked about a couple weeks ago, is present at the base of this tree. Remember, our earliest ancestor is protozoans, when we came from protozoa. Then from there, we start to move up in complexity. So we start the Humatozoa, which includes everything but our Porphyria, so our sponges, but it also includes ctenophores, Plantozoa, and Nadaria. And then we have another group, and then we break off into some other categories that we'll dive into sort of over time. But this is our broad picture of animal phylogeny as a whole. A lot of diversity, lots of big groups, something that's been studied for a really long time. Taxonomy of animals, as you'll recall, dates back to the 1800s with Linnaeus, right? That genus species we applied to our name, right, Homo sapiens, that dates back to the 1800s with Linnaeus. So this tree has been developing for a really long time. It's the best as it's ever been, but again, it still requires quite a bit of revision and understanding. And again, it continues to go all the way out to we get to the deuterosomes, which include our chordates, which is us, and then our starfish and radial and things like that. So lots of things fall under our lovely protozomes, but again, we fall under the deuterosomes, being arguably more complex. But that's just a human saying that a human is more complex than a starfish. Right. So, all right. So that's our broad view of some of the key developments of animals, broad view of the phylogeny of animals. So let's dive into kingdom Animalia. As I defined, this is just the metazoans. That's us. We're the metazoans. We share a common ancestor for everything on this tree, right? So we're all grouped broadly as animals. Animals is just things that are cute and fuzzy, right? Animals include the creepy crawlies and the worms of the world, right? But this is our kingdom Animalia. There are two distinct groups, the Parazoa, which are animals lacking tissues, therefore organs, and have a definitive symmetry, which is going to be our phylum Porphyria, sponges. And then the Humatozoa, which are animals with a definite shape, symmetry, tissues, and potentially the presence of organs in organ systems. So two big groupings, Parazoa and Humatozoa. So let's start off with the most basic, all the way here on the far left of the phylogenetic tree, the Parazoa or phylum Porphyria. All by themselves, all 26,000 species of sponges. They're among some of the most abundant animals in the deep ocean. They're very common off coastal areas as well. So if you go to a place that's not polluted really heavily, you can find sponges pretty much everywhere. In particular, when you go to the tropics, sponges are everywhere. Let's go. You've probably seen a sponge, you just probably didn't know it was a sponge. You know, if you guys ever bought like a natural sponge at the store, like those those like the Loofahs, that's an actual living sponge at one point. Just testing them. So what do sponges look like? Well, here's a nice handy dandy picture of what a sponge looks like. Now, they lack a level of symmetry to them, to their body shape, but they do have a couple of distinct things about them. Now, what do I mean by they lack symmetry? Clearly, this middle part here of the sponge has symmetry, but clearly there's no symmetry on this entire sponge body, right? Not like us, right? There's no level of left and right. There's no level of circular symmetry. There might be circular symmetry here, but again, across the whole body of the sponge here, that simply doesn't exist. Now, sponges have larval free swimming stages, followed by adults that remain sessile, i.e. non-motile, attached to the surface. And they are truly multicellular. They kind of just look like giant tubes, but that's just really all they are. And as we discussed, they have those 3 functional layers in their body. So the 3 functional layers in our sponges are the outer epithelium. Basically, that's the outer area, as it implies, epithelium being skin. Right? Your outer epithelium is your skin, right? Mesohyl, which is comprised of 3 things. The first is the middle layer, which is basically a gelatinous matrix. Think of it like Jello. Sphericals, which are needles of calcium carbonate, giving the sponge a level of structure. And then the spongin, which are reinforcing tough fibers of protein, again, giving the sponge structure. There's actually a really cool sponge out there. When it dies, it leaves its calcium carbonate shell out there, and it looks like just like a tube, a hollow tube, but it looks like a skeleton. It's really wild. It's called the ghost sponge. If you have a few minutes, you should Google ghost sponge. It's really cool. And then they have the lining of their internal cavity. Now, within there, we have cells. We have an area called the choanocytes. I never know if I'm saying all these right, but I assume that's what you say it, which are what we call collar cells. They're flagellated, which contributes to water circulation. But it's flagellated not in the way like for motility. It's flagellated just to flap, right? That's what flagella do, right? They flap. They also face the internal cavity, and they engulf and digest food from passing water. And to put what a sponge looks like together, this is it. I hope when you see this, you look and go, oh, that's really simple. That's all it is. All a sponge is a tube. And I hope that sort of, thinking about how complex you are, when you look at that, you go, it just looks like a sock. A living sock, basically, right? That's really all a sponge is. But again, we have this inner tube, which is basically its inside body. But it's not an inside body in the sense of like your inside body. It's in constant contact with the outer world. And so we have these choanocytes here. You can see all the structures of the sponge here on the inside as well. But again, that's all we have. This tube with cells that interact with the world. It has the flagellum that help keep the water moving, whipping them up so that the sponge can ingest things that have decided to flow into its cavity. Remember, they are sessile when they're adults. So they have these flagella to flap in the wind, stir up an air current, bring food to them so they can digest it. Why? They're not photosynthetic. All animals are heterotrophic. So they flap these flagella, create a nice current, to suck in dead stuff, living stuff, so they can ingest it. Okay. This is our sponge. And I hope you can see why this is the most basal of all the animals. It's just a hollow tube. That's all it is. A hollow tube that ingests. Now, sponges have 2 capacities for reproduction. The first is asexual. They can fragment their body, either on purpose or not on purpose. And then those body pieces that come off can go form a new sponge. So picture like this, I chop my hand off, I throw it out the window, I come back a week later, and there's a new patchwork down there. That's what they're doing. I know, you guys are probably like, ooh, definitely don't want another professor occurrence. Doesn't sound fun at all. So that's their asexual reproduction. Then their sexual reproduction. So these choanocytes here actually take their flagella, modify their body structure, their cellular structure, and become sperm. So that's kind of cool. So they take a structure that they already need for feeding, and they repurpose it into a sexual cell. Sperm. Now this sperm is captured and passed to an egg in their mesophyll, which can then develop within a mother or in open water, and then it produces larvae. So just a pre-form of the adult stage, which is motile and will eventually move around and settle, land on a hard spot, and form this adult sponge that we see here. So that is our sponges. Again, hollow tubes that eat whatever come in contact with them. They reproduce sexually, but typically they reproduce with themselves, changing these into sperm, fertilizing their own. Again, they do have the capacity to fertilize other sponges, but within sexual reproduction is actually pretty common. So that's the basal groups. Let's move on to the more complex, which is the Humatozoa, which are animals with true tissues. This is not tissue. This is one solid mass of cells. There's specialization of the cells, but that is not true tissue. The Humatozoa have tissues. Now, They have, and again, there's no symmetry in our sponges, but the Humatozoa have radial and bilateral symmetry, as we discussed. And they have embryos with distinct layers, the inner endoderm, the outer ectoderm, and the middle mesoderm. And again, these are tissues we spoke about 10 slides. So let's talk about, probably one of the more basic of those, which is the Phylum Tinafora. The Tinafora is comb jellies, sea walnuts, or sea gooseberries. That's my favorite, sea gooseberries. Now, they have 8 rows of comb-like plates that are fused cilia that beat in a coordinated fashion. What does that actually mean? Tentacles. That's all. That's the, if you ever wanted to embellish the way you say tentacles, just say that. Not like you ever would, but that's an embellished way of saying tentacles. Right? You guys have all seen the jellyfish before. Jellyfish have tentacles. All they are plates fused that are fused cilia that beat in a coordinated fashion, all tentacles. Many of these are bioluminescent, so glowing, using the production of luciferins, which are bioluminescent proteins, to glow, producing a pretty sweet color, doing some pretty interesting ecology things. Now, their tentacles, 2 of their tentacles are covered what we call colloblasts, which discharge a very strong adhesive that is used to capture prey. And again, as I mentioned, their phylogenetic position is actually still unclear. So these are not true jellies. These are what we call comb jellies. We'll get to the true jellies in a moment. But these are, again, Tinaphoras, not true jellies. They are comb jellies. Now, in terms of the phylogenetic tree of life, again, we're moving up in complexity. That's sort of the way this lecture proceeds. We're kind of moving up in complexity until we make it to the most complex, and again, the most complex is animals, right? Chordates, things with spines. Nothing we have thus far has spines. So that's our comb jellies, pretty cool group. A little bit unclear where they fit in phylogenetically, but let's move up to the next more complex, which is the Phylum Nadaria. One of my favorite phylums out there, and it's kind of an odd phylum. It's primarily marine, but there are some freshwater Nadaria out there. They are dipoblastic, meaning they only have 2 distinct tissue types. And their bodies have no distinct tissues, but no, I'm sorry, they have distinct tissues, but no organs. So they have no reproductive, circulatory, or excretory systems. Kind of a weird thing. And they don't have a concentrated nervous system, not like the way we do, or other things we'll talk about which they do. And they capture their prey, what we call Nemo Cysts, which are unique to the phylum. And so this is where our Nadaria fits. Again, they're still Humatozoa, but they're of the more basal ones. And there are 2 basic body forms of our Nadaria, which, again, it's going to seem weird, but again, when you think about the underlying biology here, between these 2 very, very different groups of organisms, it makes sense why they're grouped together. I hate that they're grouped together, to be honest with you, but they are. So Nadaria encompasses corals and jellyfish, true jellyfish, not comb jellyfish, but true jellyfish. Again, you would never think Great Barrier Reef corals and then a jellyfish in the Great Barrier Reef are the same phylum, that they're so closely related phylogenetically, but they are. I hate it, but it's the way it is. So they have 2 distinct types of body forms. Polyps, which are cylindrical and sessile. This is what they look like. They have that outer epidermis and this overall structure here with the mouth. But it's sessile. They're anchored down here at the base. And then they have the medusa, which is actually just a motile form. It's umbrella-shaped. It's free swimming. So you think like, hey, this is what a jellyfish looks like. That's what a jellyfish looks like, right? You can see it's got its mouth. It's got its gastrovascular cavity. So it does have that level of tissue, right? That's specialization, right? It has a mouth, it has an epidermis, it has like epidermal layer in its gastrointestinal tract as well. So again, moving up in levels of complexity here. But again, 2 basic body forms of a Nadaria. Again, you wouldn't imagine corals and jellyfish are closely related, but again, they're related because of how similar their biology is with one another. This is all coral looks like at the microscopic level. Kind of a weird thing. Now, there are some commonalities up to their body plans which group them together, including the way they exchange gas, their site of digestion, how they discharge waste, and their formation of gametes will vary, formation of gametes as well. So again, lots of things that make sponges, corals and jellyfish are very, very similar. And they have 2 layers to their body wall, as we saw. the outer epidermis, and the gastroepidermis. I'm sorry, the gastrodermis here as well. So they have lots of similar structure. And again, they don't look like they'd be related at all. But again, these similarities make them related. Now, the support for their body is basically done via their gastrovascular system. Basically creates a hydrostatic skeleton, providing rigid structure for them. We don't think of jellies as having rigid structure to them, but if you've ever squeezed a jellyfish before, you know that jellyfish have, have some strength to them. You can't just squash a jellyfish, right? You press it and you feel it contracts and has some strength to it. Why does it have strength? Because of that gastrovascular space. Now, many of these species also build an exoskeleton of chitin and a shell of calcium carbonate around them. Remember, the polyps are the sessile form. So when you think about coral, they have all this biology that we say, but they surround themselves with that very thick calcium carbonate shell. So if any of you have ever been to the, say, New England Aquarium, or if you've been to like the tropics where there's large coral, you know coral are basically like rocks. But that rock is not rock. It's calcium carbonate, but it's living tissue on corals. Doesn't know. So just as a note, if you're ever in a tropical area and you see someone messing with a coral, tell them no. Why is that? Well, one thing people like to do with corals is they like to carve their initials in corals. But the functional equivalent of that for you would be if someone took your shirt off, took a knife, and carved their initials in your skin. So if you ever see anyone doing that to a coral, smack. That's, again, like carving someone's initials in your skin. Not like tattoo carving. I'm talking about like knife carving. So, but again, it is living tissue. It might look like rock. It might be as hard as rock, but again, it is living tissue. So that's my shtick about being next to corals. They're Nadaria, but it's living tissue, even though it looks like rock. So Nadarians have an interesting life cycle. So many Nadarians only exist as polyps. Others exist only as medusa, but many alternate between these two forms. But both phases, either polyps or medusa, consist of diploid individuals. Haploid is not really a thing. I mean, it is for the gametes, but in terms of body structures, we're always talking about diploid organisms for animals. It doesn't know. Whereas their plants have that sporophyte and gametophyte generation, animals really only exist as adults in diploid shape. In diploid in terms of the genome, just as a note. Now, in generally, the species have both a polyp and medusa in their life cycles, or Nadaria, but the medusa itself is what forms gametes, where we have separation of sexes, so male, female, very much like our plants. It's not like plus and minus like protists or fungi, it's male, female. We also have what's called gonochorism, where individuals is either male or female. So again, separation of sexes, that has an actual technical term. Now, the zygote, i.e. when sex occurs, when sexual reproduction occurs, it develops into a planktonic planula, which then metamorphoses, so just changes structure, into a polyp. Then the polyp produces the medusa or other polyps asexually. The sexual reproduction occurs, but once the polyp forms, it can then reproduce asexually. So, I don't know, kind of a weird life cycle, but it's just what we do. So sex is a big deal, but you can also, asexuality is also a big deal here as well. So again, the function here would be like, you make a baby, and then the baby forms brand new babies from itself. That's basically what they do. Kind of a weird thing, but so everything's always weird when you put it in human context, but for them, it's just average, the facts of life, right? Now, the big evolutionary invention for the Nadaria is digestion. Sponges, they just absorb stuff from the water and they secrete it out of cells, right? They basically do what your cells do. But the Nadaria have that gastrointestinal tract, not a full one. It's only got one opening, as we saw. But again, the first gastrointestinal tract, we date back to our Nadaria. So the digestion itself takes place partially in that gastrovascular cavity, and then the cell engulfs the fragments by phagocytosis. So phagocytosis is just, remember, like globbing on something. Now, as we've mentioned, Nadaria have Nemo Cysts, which they use to capture prey. These are unique to Nadaria. They're distinct from the cone jellies as well. Now, these are secreted within the nemocyte, and they actually don't quite know how they do it. just as a note. And some can carry venom. So I'm sure you guys probably know one of the most poisonous organisms on the planet is the box jellyfish, right? It can kill you in like 25 minutes. Well, how does it do that? Well, it has these Nemo Cysts. So let's look at the structure of, again, this is going to be a hydra, which is one type of Nadaria. I've been sort of saying jellyfish and corals, but hydra are another part of this group as well. I just neglected to mention it. But you can see its structure here. It is the free-floating form here. It's got its tentacles, it's got its mouth, it's got its gastrointestinal cavity. But again, you can see it's got its gastrodermis here on the inside, and its outer epidermis here. But again, inside this, it produces these nemocytes, which then have flagella, which can discharge to make essentially poison to capture prey, to help them engulf prey and eat prey. So in the case of the hydra, they're producing actually free-swimming things. However, in things like jellyfish, they actually produce these nemocytes and they keep attached to their tentacles. I don't know the answer if corals can produce poison or not. I don't know that off the top of my head. No one asked me that, but usually people ask me that, so I don't actually know that off the top of my head. You could probably Google that and find out if corals produce these as well. So just so you know. So that's the basic biology of Nadaria. There are 5 distinct classes within the phylum Nadaria. Anthozoa, which include the sea anemones, most corals, and sea fans. They're solitary colonial polyps, hollow tentacles, and they have symbiotic dinoflagellates, which are protists, called zooxanthellae. Zooxanthellae are really cool. They're photosynthetic dinoflagellates. They live inside the coral's tissue. They photosynthesize for the coral, giving the coral carbon in exchange for a safe path, for a safe house. You guys have ever heard of coral bleaching? Coral bleaching is when a coral turns from a nice happy color like this or this. I know these aren't corals, but changing from a nice happy color to a bone white. Why do they turn a bone white? Well, when corals are stressed, they expel their zooxanthellae. Thus, they no longer have their color. They also no longer can photosynthesize. So that's how coral bleaching works. Then we have the cubozoa, which are the box jellies. You guys have heard of box jellyfish before, I'm just assuming. They're big boys. They look like little boxes. Then we have the hydrozoa, which includes hydra and the Portuguese man of war. You guys probably heard of Portuguese man of war. They're not jellyfish. They are in fact hydrozoa. They're really, really huge jellyfish-looking creatures. Like huge. Like they can grow like 6-7 feet wide. Crazy. We have the schizozoa, which are all the remaining jellyfish that we have not mentioned. So if you guys go off the coast of, go off to say Boston Harbor, you'll see jellyfish hanging around off Boston Harbor. And then we have the starzoa, which are star jellies, which look like this. I think they're pretty cool. But sort of interesting things like this is they look like medusa, but they're actually attached to the substrate by the stalk. So they're not the polyps. They're still the medusa, but they're attached to substrate. Kind of a weird group. These are our 5 groups. Cubozoa, hydrozoa, anthozoa, schizozoa, and starzoa. 5 groups of Nadaria: jellyfish, corals, star jellies, man of war, hydra. Pretty cool group. All grouped together by all that biology. And again, they don't look all that similar. Corals certainly look nothing like a jellyfish, but biologically speaking, reproductively speaking, they're like best buds. Like really, really simple. All right. So that's the Nadaria, that's the sponges. Again, very simple organisms, especially the sponges, right? Sponges are just a hollow tube. Now, we have the next group of organs, what we call the bilateral Kelamates, which are characterized by bilateral symmetry. Remember, bilateral symmetry, if I cut you from top to bottom, same on both sides. That's bilateral symmetry. Sponges, no symmetry. Jellyfish, level of radial symmetry. Bilateral symmetry, up and down like us, which brings us into 2 distinct clades, the protozoans and the pterosomes, as well as the ankle flatworms. These are our bilateral symmetry. So the first one is going to be the Acocella here. This is its own distinct phylum, distinct from everything else. Again, moving up in level of complexity here. So we haven't made it to the cool stuff yet, just as a note, but again, level of complexity here that, again, not so advanced, but not so basic. Now, this phylum are flat worms. Flat worms. I don't know if you woke up this morning and thought, I'm gonna hear Dr. Kearns talk about flatworms and other worms for the next 20 minutes, but surprise, that's what we're gonna do. So these are actually a really interesting group. They're actually historically speaking, were classified with the platyhelminths, which we'll talk about in a minute. But these groups actually have a very primitive nervous system. So again, moving up that ladder of complexity. And they also lack a digestive cavity. So kind of a step backwards from the Nadaria, but certainly a step upwards in terms of the nervous system. And this is what they look like. They're rather unremarkable. They're worms. As we'll see from many of the phyla, they're really unremarkable. They're just worms. But they're a really important part of ecology. So just as a note, worms, not much to say about this group of organisms. This is just what they look like. That's all they are. Which leads us to the protozoans. And I use a dragonfly as a picture of the protozoans because I really like dragonflies. They're a really cool group. Now, within the protozoans, there are either two large groups, as we've mentioned, the Lophotrichozoa or the Exozoa. Now, as you can notice that the Lophotrichozoa have many, many more phyla than the Exozoa, but that doesn't mean there's more species here versus species here. As you'll see, the Arthropoda, the most speciose group on the planet. So just because you have more phyla doesn't necessarily mean you're more diverse. Now, let's start off with the more basic ones. These are the Lophotrichozoa. Lots of interesting groups of organisms here, including the Bryozoa, including mollusks, annelid worms, and other, again, mostly worms. That's a spoiler here. You're going to learn a lot about worms right now. Whatever. Most of them are water-bound. Water is kind of an easy thing to live in, right? Living on land is hard. But living in water is relatively easy. Most have moved using cilia or some sort of contractions of their body. Now, there are two distinct characteristics that define members of this group. So the first is a trichophore, which is free-living larva, and then a lophophore, which is a horseshoe-shaped crown of cilia, ciliated tentacles that surround the mouth used in filter feeding. I know, exciting. But we have the trochophore here, and we have the lophophore here. Mouth, anus, gut, trunk, anus, band of cilia, mouth here. Again, what doesn't look so exciting, but there's a mouth and a butt. Have we had a mouth and a butt thus far? Nope. I know, that doesn't sound like a major evolutionary innovation, but a full-on mouth to butt digestive system, this is where it first came to be. We take it for granted. But most of the stuff we've talked about thus far, well, actually everything we've talked about thus far, poops out of its mouth. Because its mouth and its anus are the same thing. But now, in our friends here, we finally have a mouth and a butt. We have distinct organs, right? A fully enclosed gastrointestinal tract. Now, is the anus and mouth really close here? Yeah, but there's still a full-on gut. Now, exozoa contain animals that molt. So they have an exoskeleton, so a hard external skeleton, primarily composed of the polysaccharide chitin. Because of how abundant the exozoa are, chitin, which makes up these exoskeletons, is actually the second most abundant macromolecule on the planet, only after cellulose. So picture how many plants there are. That's how much cellulose there is. A picture a very close number to how much cellulose there is. That's how many arthropods or exoskeleton-containing organisms there are. A lot. Now, there are two large groups within the exozoa, the arthropods and the nematodes. One is cool, one is lame. So, just as a note. So, within this group, we're going to actually talk about, we're not going to talk about all these groups. We're going to talk about one of the more interesting groups with the platyhelminths. I can never know if I'm saying that correctly, but that's how I pronounce it in my brain, which are flatworms. Now, they might not seem like a pretty, important group, but they have very simple bodies. They have no circulatory or respiratory system, but they have a complex reproductive system like we do. Not exactly the same as us, but getting more complex. As opposed to just like spilling your material out into the environment, there's a level of complexity here to sexual reproduction. Now, there are many groups of marine and freshwater of them, and the best known is actually a group called the Planaria. Planaria are pretty cool. They're used as model organisms to study tissue regeneration. So, for instance, at Tufts University, across the river in Medford, there's a Center for Regenerative Medicine. They use Planaria to understand how potentially we can regenerate human limbs, to regenerate human organs. So again, flatworms might seem insignificant, and again, you'll see what they look like. This is a Planaria. Looks really boring, but really important medical use. Now, in this group, we also have parasitic flukes as well as tapeworms. You guys probably heard of tapeworms before. I was browsing the internet the other day. I went down a rabbit hole learning more about tapeworms, and I watched a video of a doctor pulling 26-foot-long tapeworm out of someone's mouth, because apparently it could not be pulled out through their butt. Like 26 foot long. Imagine that. Like a clown pulling it out. It was gross, but it was also pretty cool. So, now, flatworms, as we see here, you see it's got eyes. These eyes, as we'll learn, they're not true eyes. They're not like our eyes, right? Seeing 3D structures, but their eyes here are still eyes sensing day and night. As you guys will learn, circadian rhythms, day-night cycles, pretty important. You need to know when it's day or night. And that's not just true for Nadaria. That's not just true for flatworms. It's true for day and night is an important separation of time. Now, there are soft bodies. They're very spongy, and they are ciliated, so they have the capacity to slide around the environment. Now, they have the capacity, they have a solid structure. Like if you push on them with your thumb, they'll break because they're tiny. But because you're talking about organisms that are maximum, like about an inch long. So they're not huge by any sense of imagination, but they do, given their size, they have some level of structure to them, and they also have an incomplete digestive cavity, as we'll see. Now, many are parasitic, but in this case down here, this is a Planaria. It's a free-living freshwater flatworm. There are also ones we find in marine environments. We also find some of them in soils as well. Now, they have only one opening to the digestive cavity, very much like we saw with the Nadarians, right? But the way this works differently from Nadarians, where they just let things diffuse out, these guys actually have a muscular system. They use their muscular contractions in what we call their pharynx. Think of it like kind of like a throat-ish region that allows the food to be ingested, torn into small bits. They secrete digestive enzymes, and then once they're done digesting it, they use that muscular system to spit it out.
What do they feed on?
What do they feed on? They feed on basically anything that's dead. Sort of what they like. So like when they keep these organisms in culture, they feed them pork. Besides the note, feed them pork. Pork has all the vitamins and nutrients they need, so that's why they feed them pork. And it's kind of gross. They feed them raw pork. But they do lack a circulatory system. So they have muscles. They have, again, one opening digestive cavity, but they rely on diffusion for gas transport. So no lungs, no blood vessels to transfer this so fun stuff around. Now the gut involves digestion and distributing food to the rest of their body. And you think about how your body breaks food down. You have a stomach, you absorb it through your two colons. Then that's absorbed into your vascular system where it's distributed around your body. They don't have that. They have a stomach that has to, using its muscles, distribute things throughout the body via diffusion. So not the most efficient way, but yeah, certainly the way they do it. Now, how do they eat? They just gnaw on things. They just open their mouths and gulp stuff in. They're gulp eaters. That's all they do. Now, tapeworms, which are parasitic flatworms, one I just told you about pulling out of someone's mouth, they like to hang out in the digestive system of, and they can hang out in other tissues as well, but they're known for first hanging out in your digestive system, attaching on, and feeding off essentially your life force, right? Your nutrients, your water, things like that. Now, they lack digestive systems altogether because of their parasitic life cycle, and they absorb their food directly through their body cells. So that's kind of cool. But this is our Planaria, so this is just a standard flatworm. They have their mouth and their butt here. They have their digestive system that's here, and they have a level of nervous system here. You can see it running up and down, what allows them to contract their muscles, allowing them to do what they need to do, move, push food throughout, crush food, get the nutrients out of the food. So this is what they have. But again, they have a level of sexual reproduction. They have their outer walls. They have this inner intestine, which runs down the center. They have their nerve cord, which I pointed out as being this sort of darker spots that are running through it here. They have, again, their mouth opening, which would come into their gastrointestinal system here. But they have testicles, they have sperm, and they have the oviduct, which is going to be involved with ovaries. So they have actual sexual organs, well, sexual structures. I don't think organs is quite the right word here. Again, this is our planaria. This is our flatworms. Pretty simple structure, but again, much more complex than the Nadaria, much more complex than the Porphyria, right? Certainly much more complex than protists. But again, getting closer to what we see as being our own shaping form, right? And we can actually physically look at this. So we can look at their excretory system, their intestines running throughout. Again, this whole mass is basically a giant pile of intestines. Then we can see there's nervous system, where we actually have a minute brain. It's not like a brain as in like our brain, but it's just involved with sending electrical signals to contract muscles to sense the environment around them. Kind of a big deal, right? Nothing we've talked about thus far has a brain. And then you can see the reproductive system, ovaries and testicles on a single individual for sexual reproduction. Again, this is new, and this level of digestive system is also relatively new. So new evolutionary innovations. And these organisms also have this really interesting excretory and what we call osmoregulatory system. Osmoregulatory meaning the regulation of water. And we don't really think about that as being an important thing, but remember, like if you couldn't regulate your water, you'd be toast. So osmoregulation is really important, not just for big things, but also for the microscopic things, right? Regulating water, because everything lives in water, is really important. So for them, they have a network of fine tubes that run through the body. They have what are called flame cells. I think it's a pretty cool name, that are located on side branches, which have flagella that move water and excretory substances into the tubules and then to pores between the cells, to which the liquid is then expelled. So they can regulate themselves, their internal water concentrations with these flame cells. Now, their metabolic wastes are excreted into the gut and then just basically spit out. As opposed to pooped out, they spit it out because their mouth and their butt is the same. Their nervous system is very simple, and they do have reproduction. Now, most planaria are hermaphroditic, male and female at the same time, as we saw, which undergoes sexual reproduction. So they have the capacity to reproduce with another planaria. They also have this capacity to have sex with themselves. They just fertilize their own egg, their own sperm. Now, some have the capacity for asexual regeneration. Now, as I mentioned, this capacity for asexual regeneration is why we use them to study regenerative medicine in humans. Now, what does this look like? Just to let you know, I don't have any slide for it, but the idea being is you can actually cut a planaria anywhere. You can cut a planaria into 5 different pieces, 10 different pieces. Each individual piece of the planaria, because of their biology, within a 2-week period, will regenerate into an entire brand new planaria. So you're thinking about, I chop a salamander's limb off and it regenerates a limb. Yeah. This is, if I chop the salamander into 5 pieces, each piece would develop into a brand new salamander. So planaria take what salamanders do and just up the ante. Again, you can take them, cut them apart, and they'll regenerate into 5 pieces, 5 brand new planaria. No. They can do parts, but not this level that planaria can do, which is crazy, right? Just picture chopping yourself into 5 pieces, and then 5 brand new you pop up within 2 weeks. It's crazy, right? So now there are 2 major groups of our flatworms: the free-living Tubularia, which you know, they are what they are. They're freshwater. They're probably not monophyletic. The Dugesia is a pretty common planaria used in biological labs. So what they use over tufts for regenerative medicine is the Dugesia. And that's our free-living ones. And then we have the parasitic ones, which are the Nematodes, which are flukes. These are the ones that are parasitic, attaching to humans' bodies. This includes some pretty relevant pathogens, including schistosoma, oriental liver fluke, which is caused by this organism, and several others, again, including our tapeworms. Lots of things fall within our flatworms, But we can think about how a parasitic worm might get around. Well, if it's coming to us, how do we get a parasitic worm inside of us? Well, we have to ingest infected tissue, right? Fish, pork, things like that carry these worms. So it's always, the worms I'm talking about are actually cysts. So they form these cysts that allow them to kind of hang around so that it can then be passed to an organism, pass through their stomach, and then infect that organism. So cysts are like spores of bacteria, really hardy structures that allow them to survive really crappy places like guts. But again, parasitic life cycle. So if you're ever worried about getting like a worm, you just have to cook your food. Eating raw fish or undercooked pork or poultry, well, not really poultry, but like lamb and stuff like that, is the best way to get one of these. So as long as you cook your stuff, you're okay. Cysts are tough, but they're not like cook at 300 degrees tough. Just has enough. Now the schistosoma, really important trematode in human health, causing blood flukes called schistosoma or schistosomiasis. It's a really common disease. Afflicts about 200 million people, Asia, Africa, Latin America, and the Middle East, so tropical areas, warm areas. Every year, about 200,000 people die from these phylum. So medically very important. 200,000 people is not an insignificant amount of people, especially when you have 200 million dying, getting infected. Just think of how taxing a parasite is in your body. Right? So you're thinking like, just as a note, like this level of infections is like infection levels like that we see for malaria. Really, really high amounts of infection. So really bad problem for the tropical areas. But the way these work, for those of you that are interested, the fertilized eggs have to make it through the wall of your intestine into your blood vessels, or the urinary tract to eventually get out and spread as well. So parasites inside you, I know it seems kind of gross, but again, really important to the tropical and warm areas of the world. Lots of people die. So this is our tapeworms. So these are the ones that you guys have probably heard of before. I shouldn't have to say this, but a lot of people try to take these type of tapeworms as a digestive aid. Do not do that. That is so stupid. Don't ever do that. But just letting you guys know, this is what the front end of tapeworms look like. They use these hooks to burrow into your intestinal wall, hooking on, sapping your nutrients, your water, and all that fun stuff. Again, can't stress this enough. Don't buy tapeworms for a digestive thing. You will suffer severely. They're parasitic. They sap your nutrients. Not good for your body. Because eventually, if they get big enough, someone is going to have to pull them out of your mouth, or they're going to spread to your other tissues and kill you. I see stories like this on the news all the time. Oh, I bought a tapeworm from Mexico. It's really bad. It was good. So just as a note. Now, most of the tapeworm body is comprised of plogloides, which is basically a complete hermaphroditic unit containing male and female parts. So the way a tapeworm grows, once it attaches to you, it just basically adds segments called progloides. Segments and segments and segments. Now, for a tapeworm to reproduce, if it wants to get out of a human, it just sheds one of those segments. You poop it out. If something touches that poop, they can pick it up and become infected with the tapeworm. The tapeworms spread relatively easily, provided you touch poop. Now, in case of, so we've been talking sort of things about medical ones like this one, but there are also ones that infect cows. So in this case of the beef tapeworm, it's a common human parasite, but it's actually commonly found on beef tissue. So once you get a tapeworm infection long enough, it eventually passes into your muscular system. And again, this is our tapeworm biology. It has the hooks, it has suckers, where it's drawing nutrients from your intestine, but this is how it anchors itself. Again, you can see how it anchors itself. Individual segments that has segmentation, called the progloides, which has both structures. One of these pieces can break off. You can poop it out and give someone else a tapeworm infection. And they're really, really small. Like these little segments are very, very small. So it's easy to like say you had a tapeworm and you wiped your bum, you didn't wash your hand, spread it to someone else by touching it. Besides that. So that's our tapeworms. That's in-depth look at creepy crawlies inside your body. Hope you guys enjoyed that. Now we're going to move to the Phylum Rhodophara, or the rotifers, which is have bilateral symmetry, and they have unsegmented bodies as well. Now they have very highly developed internal organs, and they're also called corona, not like the virus, but just like or beer, but that's just what they're called, corona or wheeled animals or wheel animals. So What does that mean? Well, they have this ring of cilia on their anterior end, back end, and they're used for locomotion. So what does a rotifor look like? This is what a rotifor looks like. So you're talking very small organisms, right? This is 14.5 microns. So this entire organism is about 150 microns or so long, not very big. So it's microscopic. Multicellular, has organs, but still microscopic. So look and see what we have here. We have a mouth, we have an anus. So it has a full-on closed digestive system. Doesn't poop out of its own mouth. We made it, guys. We made it. Now, you see it has a brain. It has its pharynx. So think of it like almost like a throat sort of area. It has the digestive tract, and it has toes. Not like toes as in our toes, but like that's just what they're called, toes at the end. Again, used for propelling itself through water. Same way your feet are propelling yourself around, right? Your toes push off. Their toes just push off the water. So that's our rotifers. I don't have anything to say about them again, except for their full-on digestive system, their distinct organ. They're cool organisms, by the way. I can talk for an hour about rotifers, but we're gonna leave it there to move on to a phylum you've definitely seen before, Phylum Mollusca, which includes slugs, snails, clams, and the weird group being octopi and squid. I don't know about you guys, but when I was growing up, I never thought an octopus and a clam were really closely related. Nor did I ever think a slug and an octopus or a slug and a clam were also closely related. They just don't look alike, right? But as we'll see, structurally and biologically, they're really similar. Some have a shell, as in clams and snails. Others simply do not, like octopi. But these are our mollusca, Phylum Mollusca. So we can see just some groups. The beloved octopus here. Oh my goodness. Forget what the one on the left is. Sorry. The octopus, he's looking at you. He's giving you like, why don't you pay attention? Then we have a nautilus here, and then we have a banana slug. It's actually what it's called, a banana slug. It looks like a banana. They're pretty big, as you can see. So again, all these different things comprise the mollusks. Again, you wouldn't look at them and say, wow, these are all similar, but they are. It's like the Nadaria, right? They don't look similar, but let's talk about what makes them similar. So they range in size. They can be microscopic to huge. I'm sure you guys have probably heard of, well, maybe you haven't, but there are things called giant clams. Giant clams can weigh over 500 pounds or over 250 kilograms, for those of you that like the metric system. Huge, right? From microscopic to the size of a truck, basically, right? Like a pickup truck. Huge. They evolved in the oceans, and most groups have remained there, but there are some gastropods, as we know, were slugs and snails that live on land. Some also live in freshwater. But most of this group of mollusca live in the ocean. And they are a really important source of food and important economically. We know pearls are of value, right? Pearls have economic value, right? You can sell them, buy them, right? People like them. We also know that pets, in case of the zebra mussels, are really important, really abundant source of mussels for tanks as well. We also know many people have in their aquariums, they have like snails and stuff like that. Again, falling under the phylum Mollusca, and also other things we just eat, right? We eat mussels, we eat snails, we eat abalone, we eat all these things. So economically important. Not like the other things we haven't talked about don't serve some role, but these ones we actually directly harvest from nature, have like a direct value. So if I kill an abalone, I could sell that abalone. And again, this is what we have. Pearl producing clams. We can get pearls out of them, sell them. People like them for whatever reason. I don't know. I'm not a jewelry person. Now, their body plan is similar. Again, it might not look structurally like they're the same, but again, they all have the same body plan, like the Nadaria have the same body plan. They have a mantle, which is a very thick epidermal sheet that's bound to what we call the mantle cavity, and that secretes a shell if there is one. Remember, octopi don't have shells, slugs don't have shells, but they do have a mantle. Then they have the foot, which is the primary means of locomotion, and it's divided into arms of tentacles in what we'll term as our cephalopods. And the foot can also be used as like a extend out sort of thing. Now, they have internal organs. So again, moving up that channel of complexity. The coelom is highly reduced, so it's limited to very small spaces around the excretory heart and other organs. So we've got a heart now. We've made it to the complex stuff, right? Now, the digestive, excretory, and reproduction organs are concentrated in what we call the visceral mass. And then some of them have, many of the aquatic mollusks have what we call cnidia, which are just the gills. And for our bivalves, so things like mussels and clams, they also are used to filter food. So many of our bivalves, mollusks, clams and mussels, filter feeders, filtering out food from the water. So this is what we have here. So we have two distinct groups here. We have the chitons, which are going to be our shelled organisms, and our gastropods, slugs, and snails, and things like that. So let's look at these organisms. So we have the mantle, and we have that outer shell. We have the gut, so complete gut system, mouth and anus. We have other internal organs here as well. We also have gills. So again, stepping up in complexity from what we've seen before. Also, there's a nervous system as well, and a circulatory system, as you can see here, heart. Now, our gastropods, so the example I have here is a snail, but this applies to all gastropods. They have antenna, which is a new organ for us. Antenna, as you guys have probably heard, little things sense the environment around them. Eyes are not really common. They have them, but they're not very good. Antenna are pretty awesome. Antenna are great for sensing sense all sorts of things in the environment. So antenna are a new thing here. Now we can see the mouth going into the digestive system, and then we can see the gut, and the bum is down here somewhere. But we can see all these different organs, including lungs for those that live on land, like snails do, and slugs. Now the important thing here is you'll see this as a gastropod with a shell. Now the shell itself serves a purpose. It's not just for like protection, right, for snails, but it actually houses their organs. So people like to think of like these shells are just like protective and no, they house organs. It's a part of this living organism. So like corals, right? Corals, they might seem hard. Same way these shells seem hard, but they're actually just part of the physical organism's biology. So just as a now we can look at some other. So we have our chitons. So like many oysters and many other types of things. We have our gastropods, slugs, snails, things like that. We have bivalves such as mussels. Again, you can see all these structures. The mantle, we have muscles, we have guts, we have shell, or we have all these systems, complex systems, including the foot, which we haven't really seen thus far. They're technically in a slug here. The foot is the entire body, like just pulls itself forward. I don't know if you guys have ever seen a mussel or an oyster just like kind of doing its thing, but mussels and oysters can extend this foot out. It looks like they're sticking a tongue out and capture stuff in the environment or pull themselves forward if they want to. I don't know if you guys have ever seen that. I love mussels and oysters and stuff like that. I'm a big shellfish guy. But again, it's weird to see them stick up his tongue and pull something in with it. Kind of funny. So those are our bivalves, and then we can have our cephalopods. And one of the common things people do in labs is dissect these, but there's kind of interesting things here. Now, our cephalopods, including squids and octopi, you can see they have their mantle, they have the guts, but they have a couple of interesting things. Now, they have these tentacles and arms. Now, cephalopods are a really interesting group. They have very, very complex nervous systems. And I mean that. Their level of complexity of their nervous system is not as like amazing as ours, but it is incredible. And what do I mean by that? Well, they have a brain in here. But the cool thing about their brain is only about 30% of their brain would be actually in their cranial cavity. The remaining 70% is spread out among their tentacles and arms, giving their tentacles and arms the capacity to move independently of one another. Basically, think about if your brain was 30% here, and then the rest of it was in your limbs, meaning your limbs could do whatever they want without control from the central brain. That's what these guys have. Really cool, really important. So definitely recommend if you have, I don't know, 20 minutes of your life, dive into the neurobiology of cephalopods. Really fascinating. So for those of you that ever heard of like octopi escaping from aquarium, because of their cool brains.
Professor, when these animals and other like aquatic animals, when they are in like aquarium, do they feel like stressed and things like that? as like zoo animals, they can feel like stress or depression for being like not in this setting. So aquatic animals are the same.
Yep.
Okay.
Yep. Everything feels stressed when they're in an aquarium setting. There's no way around it. It's just not a natural environment. Right? Then you have all the pictures and lights and all this artificial stuff that's very, very stressful for every organism that can sense it. So let's talk about the mollusk body plan. We'll talk about the shell. The shell, as you guys probably know, very, very hard. Helps protect them from the environment, helps protect them from predation. So it's secreted by an outer surface of the mantle, and it's not essential, right? It's not an essential function for this group of organisms, but it has repeated loss as well as reduction across the mollusk tree of life, just as a note. And it has typically 2 layers of calcium carbonate. The first is the internal layer, which actually might form pearls. There we go. Just as a note. And then pearls are formed by coating a foreign object with all the stuff to reduce irritation. So pearls, you guys ever wonder how pearls are made like farmed pearls? They put an irritating object inside the clam, and the clam hardens its shell around it, this calcium shell around it, to make it less irritating. I know it sounds awful, but that's what it is. So that's how pearls form, in case you haven't learned. Now the body plan is what we call radula. So this is characteristic of most mollusks. They have rasping tongue-like structures that are used in feeding. I know it sounds awful, but they're used to scrape algae. In predatory gastropods, they're actually modified to drill through clamshells. Now, what is that thing? What do you think about that? Well, it's actually the way it sounds. It's actually like drilling into shells. Like you vibrate it back and forth to get into shells. I know that sounds crazy, but it's really what they're doing. Now, there are some groups, including the Conus Snail, which is among some of the most poisonous genera of organisms on the planet. Their radula has actually been modified into a harpoon with a venomous gland. So they can just essentially shoot a harpoon at a prey, inject venom, essentially killing the prey instantaneously. Kills us very fast too. Just as a note. And then our bivalves actually don't have the radula. They use their gills in feeding. But yeah, oh yeah, there's lots of stuff in the tree of life that can kill us. So think about, so just to sort of take a step back, there's lots of things on the tree of life that can kill us because their poison targets common structures, respiratory, circulatory systems, blood, all shared by us. It's slightly different, but again, it's all shared by us. So poisons that say target a fish, which is the prey, would also target us. We just need a bigger dose than say a fish does. So we can actually physically see that here. So we have the radula here on this organism. You can see its overall outer shell structure. Kind of looks pretty cool. When I look at this, I see I don't know if you guys watched the first Avengers movie, those giant flying ships through the air. That's what it looks like to me. So we can see here we have the esophagus. We have these radial tooths, and that all encompasses the radula. And in this case, the radula is used, again, for capturing. Right. Been modified a bunch of different ways. But again, it's a common structure found in all our mollusks except for bivalves. So, clams. Now, how do they remove waste? Well, they have a circulatory system. Now, they can remove nitrogenous waste, so waste like with nitrogen in it, so ammonia principally. This is found in, this is what we call Nepherdia. It consists of cilia-lined openings, which we call that. I'm not going to try to say it. You can try to say it on your own. And they have tubes secrete things into the mantle cavity, which then is excreted into the environment. They also have circulatory systems. It's an open circulatory system, so they have hemolymph. Hemo, as in blood, but not quite what we have. Not hemoglobin, hemolymph. Now, hemolymph will come up again, but what ends up happening is hemolymph is essentially just a primitive form of blood. This is what they have. This is what insects have. It's yellow in color. It is kind of a cool thing. It's yellow in color. What it does, it's not really like pumped, but it basically essentially sloshes around their hemocell, which is basically like their circulatory system, and it goes into their 3-chambered heart. Now, that's commonly what we see, open circulatory systems. But cephalopods actually have a closed circulatory system. Now, how do they reproduce? How do they get it on? Well, they're actually what we call gonochoric. So the individuals are either male or female, like us. There are some that are hermaphroditic. Some oysters have the capacity to change sex, so transgender. So that's not quite the right word. Transsexual, probably the better term to use there. But they have some pretty cool strategies, right? So the capacity to change genders or sex is not just a human thing. Pretty common across the tree of life. So, now, most engage in external fertilization, i.e. male meets female, sperm and egg, bada bing, bada boom. However, gastropods can have internal fertilization as well. And as we've seen for all these deuterosomes, the mollusk zygote undergoes spiral cleavage, so you build on top of what you already have. Now, their life stages, as we've mentioned, they have two distinct life stages. The first is the Trophophore, which is the free-swimming larval stage, and the Villager, which is the second free-living larval stage that's only found in bivalves in many marine snails. But both forms widely drift in the ocean before they become the full-on adults that we know. So this is a mollusk trichophora larva, and this is a gastropod Villager larva here, again, swimming around, floating before they become a full-on adult. So they just develop, right? So you can, it's not like in the sense like I'm a kid, then I become an adult. It's not that level of development. It's like a complete metamorphosis. Right? If this is going to be a gastropod, that doesn't look anything like a gastropod, right? Thinking like a slug, right? But that's going to metamorphose or completely rearrange its body structure to become gastropod. Now, there are 7 classes, 8 depending on who you asked. Again, it depends on who you asked. There's a guy at UMass Boston that actually is like one of the world leaders in this area. And if you ask him, he says 9. It just depends on who you ask. As I always mention, taxonomy is kind of a sticking point for most biologists. But I'll mention 4 of them here. And they're the Polyplatyphora, which are chitons, gastropoda, bivalva, and cephalopoda, all of which we've sort of explored today in some shape. So the chitons, they look a little bit like this. They're marine mollusks that have oval-shaped bodies with 8 overlapping dorsal calcareous plates. The body is not segmented under the plates, it's just these plates are segmented, and they are what we call grazing herbivores. So they have the capacity for movement. They slide around on the ground. Gastropods encompass our limbits, snails, and slugs, primarily marine group. There's also marine and terrestrial mollusks, gastropods, as you know. You guys have probably seen snails or slugs hanging around your garden or outside at some point. They're gastropods. They sometimes have a single shell. There's some, as you guys probably know, there's many slugs out there that don't have a shell. But a lot of gastropods do have a shell. Now, their heads typically have antennas. A pair of tentacles that have eyes, which we would typically call antennas. But they're technically modified tentacles here. They're not full-on antennas, as we'll see in the arthropods in a moment. They also have what we call a torison, which is actually unique to this group. It's actually a mantle cavity in the anus are actually moved from posterior to the front. So instead of being, picture like, instead of your butt being here, your butt's up here, and your head's down here. That's what they have. I know, it's kind of weird. So it's so opposite the way you would think. I know, it's kind of weird. Mouth, but weird. So another group of our mollusks are what we call the nudibranches. They're active predators. They have exposed gills, as you can see here, and here as well, that secrete very distasteful, like slightly poisonous chemicals, and they can actually extract their Nemo Cysts from Nidarian prey and transfer them into their body structure. So remember, the Nemo Cysts are what give jellyfish their poison. So these organisms can actually harvest poisonous organs from jellyfish and use them to kill stuff. I think it's pretty cool. Now we have the bivalves, class Bivalvia, clam scallops, mussels, oysters, and other things. The best class, the tastiest class. Mostly marine. There are some freshwater mussels and scallops and clams as well. Not super common.
What do mussels eat?
Mussels are filter feeders. So that's the common here. So water enters through their siphon. So just their essentially like an opening. And exits through their excellent siphon, but it doesn't do that before passing through the gills. What do the gills do? Well, the gills absorb oxygen, but the gills also trap particulate. What's particulate? Just dead stuff in the water. So they are what we call filter feeders. They pull stuff that's hanging around in the water, whatever that might be. Bacteria, protists, fungi, dead stuff, doesn't matter. They pull it out of the water, extract it with their gills. That's how they do. So they are filter feeders. They don't have a distinct head, or onto things like, say, a snail does. And they have two shells, which we call valves, that are hinged together. And they have what are called adductor muscles that counter hinge, and a counter hinge ligament that allows them to open and close. Very, very strong muscle. Like really strong. Have you guys ever eaten like ever tried to like open a mussel or a clam that's living? It's like, unless you know what you're doing, it's like trying to pry open like, I don't know, like trying to like rip wood off the wall, like really, really strong. Like picture trying to like take a car tire off without unscrewing the bolts. That's how tough their muscle is. But this is what we have for our bivalves. The adductor muscles, again, keeping it safe, cold, keeping it shelled together. But again, we see all these lovely things: stomach, mouth, heart, kidney, gills. You see the siphon here. So this is the exhalant siphon. We have the inhalant siphon. So things come in here. They're filtered via the gills, then they're excreted through the exhalant siphon. Why they're filtered. But again, they have their foot, which again looks like a tongue. And if you've ever wondered what you eat when you eat a bivalve, you eat the foot, as well as the mantle as well. You get, usually when you're eating like mussels or oysters, you usually get rid of like this goopy stuff here, and you just eat this part. Now we have the cephalopods. There are 700 species of them. They're all found in the marine environment. They are really cool active predators, and they're the only mollusks that have a closed circulatory system. And their foot has evolved into a series of arms that are equipped with tentacles and suction cups. So these are our octopi and our squid. Now, the really kind of weird thing about this group is they have jaws, but they look like a bird's beak. So if you guys remember back from our very first class this semester on evolution, we looked at the beak of the finches. Picture that, and that's what they have, an actual beak. It's weird. You can pull it out, it looks like a bird's beak. That's what they have. They have jaws that looks like the beak of a bird, and they're actually called beaks. In addition, most cephalopods, not all, have toxic saliva. Now, in terms of what we talked about thus far, they have the largest brain size. As I mentioned, octopi, crazy smart. Why? Well, they have brains that are separated across their body. Really cool group of organisms, which means they have to have a very highly developed nervous system. So we can see the underside of an octopus here. You can see it's picking up a cap. But you can see this little tiny opening here. That's its mouth. Inside that mouth, there's the beak, which you guys should just Google it and see a video of it. It like looks like a beak of a bird. It's creepy. But that's what our octopi have. Again, incredibly smart, capacity for problem solving. Now think about what that means. You think problem solving is typically something that's only reserved for humans and the smartest of animals, like dolphins and dogs. But you're talking about a very basal organism with the capacity to problem solve, to open bottles, to escape from their aquariums. Really smart organisms, really cool. So again, definitely spend some time, read up about octopi and their intelligence. Fascinating group of organisms. Now, as you guys probably know, this group actually lack an internal shell, right? Octopi and squid, they're squishy, so they don't have a shell. But they do have a couple interesting parts. So they have, actually, let me take a step back. There are nautiluses. So there are chambered nautiluses. There's a few species of those that do have a hard shell, but by and large, cephalopods lack a shell as well. And some squid and what we call cuttlefish, which are teeny tiny little squids, do have a level of an internal shell. But that external shell that we've seen thus far simply just not there. Now they have a jet propulsion system. Their siphon, they suck in water and they expel it out of their siphon. So think of it as like an extra hole to propel themselves through the water. They can swim really fast. So there are some squid that swim like, you're talking like 10 to 20 miles an hour, just by propelling themselves through water by expelling water, which is really kind of cool. They can also produce what we call ink, and actually mean that ink. So they have an organ inside them called a pen, and it looks like a plastic pen. They use to excrete ink from them, deterring predators, allowing them to escape. And they also have chromatophores, which actually allow them to change their skin color for camouflage or communication. So you can see octopi that will change their skin color relative to the environment they're in, basically blending in. So like the analogy here would be like a chameleon, right? That's probably seen a chameleon. They change their skin to blend into the environment. Octopi and other cephalopods do the same. Pretty cool. But again, we can see a relatively large squid here. Its siphon here, it's actually pretty big, because again, it's a big squid. And it's expelling water. In this case, it's pooping, doesn't know. But it's expelling gas, expelling air and water to propel itself through the environment. Now this one happens to be taking a poop, but that's but again, really cool group of organisms, like really cool. I love octopi. Sometimes I wish I had specialized in studying them, because they're awesome. They really are. And there's also cool groups of them that have the cool bioluminescent microbes in them, so they can glow and all sorts of stuff. So really fascinating group. I wish we could talk more about them, but we need to talk a little bit more about other stuff. So we have an entire tree of life to get to. Much as I love octopi and I love squid, and they're also tasty. So we have our ribbon worms, or Nematera. There's about 2,400 species. They're typically cylindrical or flattened worms. What do these groups of organisms look like? Well, most are marine. Some live in fresh water. Others live in very humid terrestrial environments like the tropics, the rainforests. Now, Linnaeus longimissus has actually been known to measure meters in length, which is actually the longest known. Which is crazy. Their body plan does resemble a flatworm, but unlike flatworms, they have complete guts. And they have what we call a rhimocele, which is a fluid-filled coelemic cavity. But this is what our worms look like. Our Nematerian worms, not particularly very exciting. Just worms. Float through the water, eating stuff, pooping, doing worm stuff. Now this leads us to annelids, which are segmented worms. So you guys have probably seen earthworms like this. These are annelids, really abundant. Some people estimate that there are well over 10,000 per acre in your average everyday forest. So clearly an abundant group, clearly a very important group of organisms. Now they have repeated units. So if you guys have ever taken the time to look at an earthworm or any worm, you'll notice they have repeating units over and over again. They have segmented bodies. So this is our annelids. Now the phylum Analytica, as you can see, it's nestled in here right next to the mollusks. But again, we haven't moved on to the most complex things quite yet. Now, what do their anatomical features look like? Well, their head has a well-developed cerebral ganglion, so it just has a well-developed brain. That's all you need to know. They have sensory organs in ring-like segments, and many have eyes, but many of them live underground, so eyes are not really a necessary component here. Now, their segments are divided internally by a structure we call a septa, and each segment has a pair of excretory organs and ganglia and locomotion structures. But each segment actually acts independently, which is pretty cool. They have a ventral nerve cord and have a closed circulatory system. And this is just an earthworm. They have their segmented body. They have a brain with their spinal cord running all the way down the ventral side. Ventral is here, dorsal is back here. Just doesn't know. But you can see they have the esophagus. They have all these blood vessels, hearts, multiple hearts. Each one has its own sort of bits, pieces of parts, intestines, nerve cord, all that fun stuff. Male gonads as well, and female gonads, so they're hermaphroditic. Most of these worms are. Most annelids are hermaphroditic. So that's our earthworms again, but annelids are not just earthworms. There's many other types of annelids out there. There's a few thousand species of annelids out there, including marine ones, which are actually kind of creepy when you look at them. If you ever go swimming like in Cape Cod, you'll pull an annelid up every now and then. They're creepy looking. They look like this, like creepy little things coming off them. They're kind of creepy. And again, they have locomotion, they have a closed circulatory system, and they have a very similar excretory system to our mollusks. Now, there are 32,000 described species of annelids at this given time. Again, probably a lot more out there. They're essentially put into two classes: the polychaetes and this Clitelia, which each have their own little sort of subdivisions as well. Now, the polychaetes include clamworms, scale worms, lungworms, tube worms, and the other one I can't remember off the top of my head. It's right there. Each have a pair, what we call parapodia in most segments, which is used in locomotion. What is a parapodia? Remember, podia refers to feet. So feet-like structures. And most are gonochoric, so they have external fertilization. So making sexy time with another organism. Now, just to give you an example of a really cool group of these, we actually have the deep sea tube worms from the genus Riftia. Now, the adults are gutless, but just because they're gutless doesn't mean they can't eat. So these organisms live down by hydrothermal vents, which are deep down in the ocean. They range from being very cold to being extremely hot. Now, hydrothermal vents are points at which our inner magma of our planet erupts from the crust of the planet. So these areas can get extremely hot, but there's no light down there, so no photosynthesis occurs. So the organisms that live down there, bacteria and archaea, are actually what we call sulfur oxidizing organisms. Stuff that erupts from our mantle, so our lava, is rich in sulfur. So bacteria have the capacity to oxidize sulfur, making inorganic compounds, so doing a form of chemosynthesis. So instead of photosynthesis, they do chemosynthesis, taking, again, the chemicals in essentially magma, using a type of synthesis, not photosynthesis, but those chemicals to synthesize organic molecules. Now, these organisms live inside the structures here of these worms, which don't eat, but provide a house for these bacteria and archaea that oxidize sulfur to produce sugar, which they then exchange with the worm. Pretty cool organisms. These worms, they look sort of nondescript. They can be upwards of 3 meters, maybe 10 feet long. They're huge. They're really cool. But again, a really cool symbiosis in a really harsh area of the world. Bottom of the ocean, there's no light, there's no photosynthesis. So we have a different type of synthesis, chemical-based synthesis. So instead of photosynthesis with this light-based synthesis to make sugar, we use the energy in chemicals to make sugar. Which again, they live inside here. You can see them. It's kind of hard to see, but they're like little purple dots inside the cells of these organisms. They take in the sulfur, they make sugar, they give it to the worms. They have this cool symbiosis. It's only made possible by the volcanic activity down here in hydrothermal vents. If you've never heard of hydrothermal vents before, I suggest you spend some time reading about them. They are fascinating ecosystems. They really are. Now we have the class Clitellata, there we go. So they have what we call Clitella. Now this is our earthworms. So they don't have a particularly well-differentiated head. So if you've ever seen an earthworm before, they kind of just look like a tube. They have a head-like structure, but they doesn't really look like a head by any stretch. They look like a head, like in a standard way you think of a head. There we go. That's what I'm going to say. So this is our earthworms here. Again, they have this middle section here, but again, doesn't quite look like they have a head at all. Now these are 2 worms getting busy, sexual reproduction. Now this is going to be a terrestrial group of them living underground. Which brings us to probably the one that will make your skin crawl a little bit, which are leeches. Leeches are found mostly in freshwater. There are very few saltwater leeches. And you guys probably know leeches. What do? Well, we know leeches mostly because they suck blood. Most leeches, however, are actually relatively normal. They just eat dead stuff or detritus. But we know leeches because, well, they suck blood. I know it's gross, but that's a small leech that you would find, say, here in Massachusetts. There are tropical leeches that can get, you're talking half a foot to a foot long. But again, the ones we find here in Massachusetts, they're annelids, flatworms, so they're not exactly the most exciting thing, but again, they rely on sucking blood, so they are parasitic in a sense, but again, most of them are detritivores. Now, the interesting thing about them, they are flattened, so unlike worms as we saw here, they are flattened as you see here. Now, they're also hermaphroditic, so they do have the capacity for cross-fertilization, as we kind of see here, right? I know this isn't a leech, but again, they have the capacity for that to happen. Now, they actually have suckers at both ends of their body. So you see the leech attached on here. It also will attack on back here if given enough time. So that's our worms. That's our annelids. Now, this leads us to the bryozoa and the brachiopoda. Now, this group of organisms, they're all basically marine. There's very, very few freshwater ones, and there's no terrestrial ones of these as well. But they're characterized by what we call a lophophore. So these are circular U-shaped rings around the mouth with 1 to 2 rows of ciliated tentacles. So it's going to look a lot like our Nadaria, but they are distinct from the Nadaria. Now, this helps them function with gas exchange as well as feeding. So this might be a case of our convergent evolution, because again, as we'll see with this structure, well, we'll get there in a second. As we'll see with the structure, it looks really similar to the structure of Nadaria, but it has evolved independently twice, thus it is convergent evolution. Now their phylogeny, as you can see, so we're sort of moving down the end, right? We talked about annelids, we talked about mollusks, we talked about these groups of organisms here. So now we have these sort of final two groups here that will round out our Lophotrichozoa before we move on up. But they're also called what we call moss animals, and they're small, very small, not microscopic small, but still very small. And they typically tend to live in colonies, the colonial organisms. Now their anus opens actually near their mouth. In their, many of them to me have this tiny chitinaceous. Chitinaceous just means it's made out of chitin chamber, which is called the zoocoeum. Sorry, the zoocoeum, not a zucchini. The zoocoeum, which allows it to attach to the substrate. And many of them will actually deposit calcium carbonate. So many of the ancient reefs that we've seen in the fossil record are actually formed by the phylum Bryozoa. But they do have asexual reproduction that does occur by frequent budding. Now, this is what a bryozoa looks like. Again, it looks a lot like a coral, right? A lot like a coral, right? But again, convergent evolution here. Structures that we see in corals, we see in the Nadaria, have evolved two independent times. Okay. But again, you see what their structure looks like. They have these lophophores, they have the mouth, the anus is right next to them, their stomach wraps around like this. They typically will secrete a calcium carbonate shell here to anchor themselves. How do they reproduce? Well, they'll bud off. This part will bud off, going onto the environment, landing on a new substrate, giving them the capacity to reproduce. Bryozoa, cool looking group of organisms. Lots of them here off the Massachusetts coast. Just doesn't have that. Now, phylum Brachiopoda, brachiopods or lamp shells. They have two calcified shells, very much like our mollusks. They have dorsal and ventral ones. They're not lateral like our bivalves, just as a note. And the lophophore itself lies on the body in between the shells, as a note. So this is what our lophophores, brachiopods look like. For those of you that have ever played like Super Mario, you guys, I don't know, if you ever played Super Mario, like the flowers that come out of the pipes, that's how I always thought these guys looked like, especially in this picture here. It looks like the flower from Super Mario that tries to eat him. But again, you can see they have that lophophore here. They have the opening here. They have a distinct mouth here. And again, they have very similar structures. But again, unlike the gastropods, which again have that hinge to them, these are really, it's not, there's a distinct upper and lower, right? Dorsal and ventral shells here. Again, but it attaches to a substrate with the pedicle or the pedicel. Now the pedicel should sound familiar. Pedicel is how plant leaves attach to the plant stems, right? So in this case, the pedicel is actually a similar structure. Obviously made of different material, but it's called the same thing. But again, all the organs that we talked about, including the adductor muscles, as we saw in the mollusks, are similar structures. But again, this is an example of convergent evolution. Similar structures evolved across multiple individuals. Wacky and wild stuff. Again, here's just another diagram of one of our bryozoans here. Just a slightly different structure. All right, so that leaves us from the Lophotrichozoa into the exozoa here, which starts us off in the nematodes. Nematodes are a fun group. They include a couple things. So vinegar eels, eelworms, and other what we classify as roundworms. They're marine, they're freshwater, they're parasitic, they're also free living. So a pretty diverse group of organisms overall. And they're pretty abundant. You don't really see them, you don't really hear about them, but nematodes are actually pretty abundant. They're pretty common across the planet. Now they have bilateral symmetry, and they are unsegmented. So as you can see in this, there's no segments here at all. But they do have bilateral symmetry right down the center of them. Now they're covered by a flexible, thick cuticle that molts as they grow. So as they grow, they just shed their old parts, getting bigger in the process. Kind of a cool thing. Now, they have a very well-developed digestive system, very much the way we do. It's called different stuff, but very well-developed, including stylates, which are piercing organs near their mouth, the pharynx, which creates sucking action. So you think of it, again, like a throat, essentially. And they have a bum or an anus. So let's look at our nematoda here. So you can see its mouth here, its pharynx here, again, for that sucking motion, all the way down in their intestinal system, down to their anus. Now, they have a couple interesting pieces here. They have testicles along the way, and they have what's called the genital pore, which is how they mate, excreting eggs or sperm through the genital pore. So they're not hermaphroditic at all here. Again, this is a cross section. Again, you can see all the different parts that you know and love. Testicles, epidermis, they have an outer cuticle that's hard, muscles, excretory system, nerve system, all that stuff. So a bit more complex than what we've seen. Not a lot, but again, slightly more complex than, say, the planaria, right? Similar types of worms, but again, much more complex organ system and structures. So they are gonochoric. Remember that sexual dimorphism, male and female, not sexual dimorphism, but they have male and female individuals. They actually have sexual dimorphism. Males are smaller than females. What does sexual dimorphism mean? Well, as we'll see later in the semester, males and females just look different. And I mean that not like how, say, like I and any other woman in this class look, but I actually mean like very differently. So pretend like I had a giant claw, and that was a standard structure of males in our species, but females lacked it. That's what I mean by sexual dimorphism, right? Or like, for instance, like elephant seals. Female elephant seals are teeny tiny. Male elephant seals weigh like 3,000 pounds. That's what I mean by sexual dimorphism. Extreme differences in the body. So, but they do have a level of internal fertilization. Now, what does that mean? Well, as opposed to secreting your eggs into the environment, well, you have to meet genitalia to genitalia for that level of fertilization to occur. So sexual reproduction is actually a thing, contact. And then finally, they have indirect development. They start with eggs, they go into larvae, and then they go into adults. Now, that's indirect. Remember, direct development is I have a clump of cells that develops into a smaller human, then develops into a bigger human, and then eventually an adult. This is indirect. They go through stages. So in this group is actually a really important model organism called Centrohabitus elegans, or C. elegans for short. C. elegans is a model organism. It's really important for genetics and developmental studies. As a note, many labs that study genetics use C. elegans as a model organism. But C. elegans actually only have 959 cells to their entire body. You can see C. elegans, they're about this big. But all of our organisms in this group have a fixed number of cells, and it's usually a small number of cells, which I think is kind of a weird thing. They just pretend there was like a fixed number of cells that every human had. We don't, but that's what's going on. Now, many of these organisms are active hunters, preying upon protists and other small animals. They are very tiny. So a lot of them actually are what we call bacteriovorous, meaning they eat bacteria. Others can be parasites of plants. Others will actually be parasitic, living in the bodies of larger animals. Now, for instance, there are nematodes that can actually attain a length of 9 meters that actually will parasitize the placenta of sperm whales. Picture that, 9 meters, 30 feet, huge. So again, 900 cells all the way up to a 9-meter organism. Huge range of body sizes in this group. So it's pretty cool. Now, nematodes, about 50 of them are known to cause human diseases, including hookworms, which can cause anemia. It's pretty common in the southern United States, as well as trichinella, which causes trichinosis, which forms cysts in your muscles, and you get the infections from eating undercooked meat. These not too difficult to cure in terms of medicines, at least here in the United States. But places that don't have as much medicine as we do, not exactly the best. But again, you can see these worms inside muscle tissue here. Nematodes, parasitic, sapping on your nutrients, making you sad. Now, there are other nematodes, including pinworms, caused by this organism here, Enterobus vermicularis, which infects about 10% of the United States population, causing itching of your bum bum. So if you ever had itchy bum bum, it might have actually been because of this pinworm. I mean, like itchy as not like just garden variety itchy bum, but like actual like you want to scratch your colon itchy. Then we have Ascaris lumbricoides, which is an intestinal roundworm, which actually infects 1 in 6 people worldwide. So it's actually one of the most common infections globally. 1 in 6, really common. The adult females can be about 30 centimeters long, but they're not particularly common in areas with modern plumbing. So places like India, which doesn't have a really modern plumbing system, inflate the number of Ascaris worldwide. Also places like Haiti that, again, not doing so well economically, don't have a great plumbing system. And there are also some serious tropical nematode diseases, including filariasis, which many of you might know as elephantitis, again, caused by one of these nematodes. Now, this leads us to the final group of organisms that we're going to probably get to today, which is the arthropods, which is by far the best and most important group that we're going to talk about in this entire class. Arthropods are the most speciose and the most abundant and the most economically important animal on this planet. And when I say that, I mean it. Every year, class Arthropoda gives our planet about $2 trillion of economic value for free. How does it do that? Pollination. So again, numerically, the most abundant, the most speciose, 2/3 of all species, and the most economically important animal that we will talk about today. Again, talk about the GDP of like European countries just for free from arthropod pollination. Super important group of organisms. There are 4 distinct and extant. Remember, extant means living. We have the crustacea, the hexapoda, and the myriapoda, and the Chelicocerata. I never can say that one right. Those are our 4 extant classes of arthropods. So what do we have here? Again, we're going to talk about all of these. We have the Chelicocerata, stuff you probably haven't seen, kind of creepy looking things, but it also includes spiders, mites, ticks, things like that, but also lots of other things that you haven't seen as well. Lots of things, but we typically think of these as like arachnids. Then we have the crustaceans, lobsters, crabs, shrimps, things like that, as well as like millipedes fall into this group as well. Hexapoda, which is our insects, bees, butterflies, flies, so on and so forth. And then we have the myriapods, which are centipedes and millipedes as well. I meant to say whirly pearlies fall into the crustacean, not centipedes. Whirly pearlies, better. So this is how it breaks down, and I love this quote by J.B.S. Haldane. He's a naturalist from the early 1920s. Haldane said, "If he exists, the creator has an inordinate fondness for beetles." And when you look at the breakdown of diversity of species, 33% of all arthropod diversity of species is beetles. There's currently close to 600,000 described species of beetles on this planet. There's close to 300,000 described species of moths and butterflies. There's close to 200,000 described species of flies. So again, beetles take the cake though. And that's why I love this quote from J.B.S. Haldane. Inordinate fondness of beetles. Just as a note, we think there's about 1.5 million species of beetles on this planet. I love beetles. That's why I love this quote. My favorite group of insects, beetles. But again, you can see other things, centipedes, arachnids, crustaceans, and other stuff. Still up the rest. But again, lots of beetles, lots of flies, lots of moths and butterflies, giving us about 60% of all arthropod diversity. So why are they so speciose? Why are they so successful? What is it about them? Why are they grouped together? Well, they are segmented. They have a head, they have a thorax, and an abdomen. Now, the head and thorax, as we'll see, can be fused into what we call a cephalothorax, but they have that segmentation of the body. 2 segments or 3 segments. They have an exoskeleton. It's made out of chitin. As I told you, chitin is the second most abundant macromolecule on the planet. Why? It's because these damn things are so abundant on this planet. I mean, they're really abundant, which protects them from water loss. It also allows them to be tough. So when you dissect a grasshopper, you're going to see how tough it is to cut through the exoskeleton of these organisms. It's so tough. And it allows them to molt. So they undergo exodus or molting. They shed their exoskeleton, growing larger in the process. And finally, they have jointed appendages. Jointed, as in like joints. Like my elbows are joints. My knees are joints. They have joints. And they can be modified into antenna, mouth parts, wings, legs, all sorts of fun. Not hands or anything like that, but yeah, we'll get there. And those can be extended or retracted. So this is what we have. So we have a bumblebee here. Well, actually, there's a honeybee. One of the most important pollinators on the planet. We have its mouth parts here. We have its head, thorax, body. Now, you can see some structures here. It's got its legs, right? They're jointed, allowing for movement. It has antenna, as you can see, has a joint, allows for movement. They have what are called compound eyes. As we'll see what compound eye looks like, there's like a bunch of little eyes melded together. Very cool looking structures. They have wings. They also have air sacs, which acts as their lungs. which are attached to a couple different things. First is spiracles, which is how they uptake air from the environment. And then they have their Malphigian tubes, which allow them to absorb the air from the environment, i.e. get it into their circulatory system. They, very much like us, have a mouth and an anus. They have a rectum, as well as guts and all that fun stuff, stomach and all that fun stuff. And the other thing I'll just make mention of about this organism is some of them have stings and poison. So bees, as you guys probably know, they have poison. Spiders have poison. So this is our arthropods. Again, this front part might be fused into a cephalothorax. Again, 2 or 3 structures, all identical in how they're done. Now they have that open circulatory system, so they have hemolymph. They don't have blood like we do. They have hemolymph, and they have a nervous system. So it's a double chain of segmented ganglia, and their ventral ganglia control most of the activities. Ventral meaning belly. And so this is what we have. This is our beloved grasshopper. Again, you can see the brain, you can see the aorta, the ovary, the stomach, the heart, the rectum, all the parts, all the bits and pieces that you know from your own biology, except for a couple special ones, right? The crop, Malphigian tubes, and the ceca. Other than that, the stuff is really similar to yours. So again, getting much more complex biologically. Now, the arthropods have compound eyes. As I mentioned, there are a bunch of independent visual units. They're called ommatidia. Again, you'll see what this looks like. This is what a compound eye looks like. We have the ommatidium here, and then we have the optic nerve here, which has to be processed by the brain. So they have decent eyesight. Not crazy good, like us or birds, but pretty good eyesight, which means they have the neuronal capacity to begin with. Now, some of them are really good, like, so for instance, predatory flying animals like a dragonfly or a damselfly, really good eyes for seeing. Others, really bad eyes. But generally speaking, better than what we've seen thus far. Now, the respiratory system is pretty similar to ours. Now, those that are marine or aquatic have gills. So if I had you dissect, say, a crayfish, you could crack open a crayfish and you'd see the crayfish has gills. Most, as you can imagine, gills are only useful if you're in water. So if you're a terrestrial arthropod, well, not really help you, right? Now, some arthropods lack any structure for gas exchange. They just allow gas to come in. They don't have any like lungs or anything to pull things in. They just rely on diffusion of gas inside their body. Now, terrestrial pods have trachea, which branch into what we call tracheoles, which come in direct contact with their cells. And then they have other parts that are connected to the spiracles. The spiracles, again, are just openings that control both airflow as well as water loss. And many spiders have a really interesting structure, which we call book lungs, which are leaf-like plates. And if you wonder how a spider breathes, it's got what we call book lungs, which look like leaves. Pretty cool thing. But this is what we have here. We have the trachea, we have the spiracles, which again are on the outside facing the environment. And then we have the tracheoles acting as essentially their respiratory system. Right? It's not carried by their blood, the air is carried by these tracheoles. Which means that if air is carried from the environment inside to the tracheoles to be distributed all over the body, that means they have to send the CO2 that they produce and all the other gases into the tracheoles to be expelled via the spiracles as well. So, now, their excretory system, so how they poop. We haven't got to peeing yet. We're just an organism that poop. It's all just one process. It's not a separate thing yet, just as a note. For most of the arthropods that are aquatic, their waste actually just diffuses through their gills, which is kind of gross if you think about it. Like, imagine like pooping through your lungs. I know it sounds gross, but like poop coming out of your nose, sort of thing. That's what they do. However, in terrestrial insects, use what we call Malphigian tubules, which helps eliminate nitrogenous waste and also allows for the efficient conservation of water. So conservation of water is kind of a big deal. So this is how they deal with their waste, either through their gills or through their Malphigian tubes. But again, this is our structure here. We can see the Malphigian tubes, again, helping get rid of waste, but they have mouth, anus. If there is gills, they're usually here. Again, can excrete waste through gills if they in fact have gills. But same structures over and over. I've showed you the same picture, just in slightly different ways for the past like 10 slides. Okay, all arthropods have this same structure. So let's start off with probably the one that you hate the most: spiders, ticks, mites, scorpions, daddy long legs, horseshoe crabs, and sea spiders. So the creepy crawlies, the things that probably make your skin squirm when you think about them. I think they're awesome, but that's just me. Now, they have the interior appendages called the Kelamates, which function as fangs or pincers. So if you've ever been bitten by a spider, you got pierced by the Kelamate. It hurts. When I was younger, I had a tarantula. You know, it's like the size of like, you know, like the top of a can of soda, a little bit bigger. That thing bit hard. Little tiny spiders don't bite hard, but larger spiders, they bite hard. So just now, their body is divided into two what we call tagmata. We have the interior prosoma, which bears all their appendages, and the posterior ophthalmosoma, which contains their reproductive organs. So what does that look like? Well, like this. Appendages here, reproductive organs here. That's what our spiders look. Now, there's an order within our, this group, I'm not going to try to say it, sorry, where there's about 35,000 species of spiders. There's this sort of note that you're never within 10 feet. You're never farther than 10 feet away from a spider. That's just kind of true. They're really, really speciose, and they're really abundant. But why are they so abundant? There's lots of insects out there to eat. So spiders, by and large, are all pretty much predatory. They hunt things down. They're good. You see a spider, you should leave it alone. They're not going to hurt you, unless you live in Australia, of course. If you live in Australia, yeah, you can kill a spider. But here in the United States, unless it's a black widow or brown recluse, you're fine. But again, they are really important as predators. If you see them, leave them alone. They're keeping your house free of pests. I mean that. Now, some spiders, as you guys know, can create silk, which is pound for pound one of the strongest fibers on the planet. Like pound for pound stronger than steel. They're really, really strong. Now, as you guys know, these spiders make webs where they catch the prey. And how do they kill their prey? Well, they use their fangs, they inject the poison to kill prey. Digest, and what their poison does is two things. It is enzymatic, so it breaks down their tissue, and it also kills them with poison. And so spiders eat by injecting their prey, killing them, injecting these digestive enzymes, and then sucking it up like a straw. I know that sounds awful, but that's what spiders do. So that's pretty cool. And so again, these are the two venomous species in the United States we have to worry about. The brown recluse, not very common. And the black widow, again, not super duper common, but these are the only spiders you should ever kill. Just doesn't know. Like, you know, this one, you can just put it outside. Most bites from these spiders are what we call dry bites, which means they don't inject poison. Poison is very expensive, biologically speaking. So most of the time a spider bites you, it's a dry bite. Just doesn't know. Same thing for most snakes. Now we have the Acari, which are mites and ticks. Very, very diverse group. Last time I checked, there's about 15,000 species of mites, or Acari, I should say. Most of them are small. For instance, like you guys have a very vibrant community of mites that live in your hair and on your eyelashes. They're microscopic, you can't see them, but they're there, they're always present. But again, most are small. They're like very much like spiders. They only have two body compartments. Unlike spiders, they actually have one body compartment where their cephalothorax and the abdomen are actually fused into an unsegmented ovid body. Now ticks, which again fall into the Acari, much larger. You guys know that they carry diseases. They like to suck blood. And these include diseases such as Lyme disease, kind of a nasty disease, and things such as Rocky Mountain spotted fever. And with that, that's going to sort of end our class for the day. We'll pick up on the rest of the things of the planet in due time.